Thanks for giving me the opportunity to share some stuff with Juventas. I'm going to go over this quickly for 5 minutes, then look forward to the conversation. So Juventas is a private company focused on using a factor-based strategy to activate endogenous tissue repair of the body. Our lead molecule is something called stromal cell drive factor 1, or SDF1. And what was shown about 15, 16 years ago was that if you could upregulate SDF1 after injury, at that time it was studied in the heart, you can induce a significant amount of repair, primarily through recruitment of endogenous stem cells, both bone marrow-derived and tissue-specific. Since those early discoveries, it's now been shown that SDF1 and its receptor CXCR4 are a key axis for inducing tissue repair in just about any end organ system. And that the mechanisms through which it works are more complex than just recruitment of stem cells. There is a strong vasculogenic impact, and the blood vessels that are created by SDF1 tend to last for months after the SDF1 signal is gone. There's also a direct effect on the tissue through anti-apoptotic mechanisms and AKT signaling. And together what this results in is an anti-fibrotic mechanism of action that helps recover scar and improve function of normal tissue. What we knew from our early studies is it was important to get SDF1 expressed in a localized fashion for about 3 to 4 weeks following a tissue injury, and that could either be done immediately post-injury or chronic several weeks or months later. The strategy we took for that was the use of a non-viral DNA vector. So non-viral DNA vectors are of bacterial origin, easy to manufacture, easy to scale up. They're interesting for our use because they provide a transient gene expression, and importantly, they're also non-immunogenic. Our lead product, JVS100, has now been in over 250 patients, has a very strong safety profile. We have been able to scale its manufacturing up to a level that has us confident that we'll meet commercial manufacturing goals and are able to do so at a relatively modest cost of goods. We have, as I mentioned, the axis is really present across all organ systems, and we've been able to take JVS100 through multiple clinical studies, peripheral artery disease, which I'll talk about, which we're currently active in a phase 2b study. We also have some very compelling heart failure data through a phase 2a study, and also dermal scarring post-surgery. Preclinically, we've been able to demonstrate efficacy in muscle regeneration, incontinence, and lung injury. What we know and we can convincingly say about JVS100 is it has a strong impact in muscle and skin and has the ability to be able to grow new blood vessels to help reperfuse injured tissue. Knowing that, where we decided to focus our lead program was in peripheral artery disease. So peripheral artery disease, huge issue, all the same reasons heart disease is: obesity, smoking, hypertension. It affects about 12 to 18 million patients in the United States, and its most advanced stage is known as critical limb ischemia. It's a field that's been primarily managed by the device industry through the use of stents and balloons. And because of the increasing prevalence of this, what we've seen is a significant decrease in this population of major amputations above the ankle. And that's great progress for the field, but it's left a significant unmet clinical need, and this patient kind of exemplifies where we fit into this. So in a very typical patient scenario, the way they typically come into the office is with a non-healing wound. That patient then gets referred to a vascular surgeon or an interventionalist, and now they can do an excellent job, as you can see taken from the top left, a patient that has no blood flow in their lower limb, to the top right, which is opening up that blood flow through the use of a stent or a balloon. The challenge with these patients is a much larger proportion of them now have diabetes, end-stage renal disease, and other microvascular complications that prevents that blood from perfusing through the foot. So despite a successful revascularization, these patients' wounds go on to get worse, and oftentimes they're amputated at the toe or foot level. And recent data out of one of our collaborating centers, Cleveland Clinic, showed that despite the presence of successful revascularization, these wounds only close about 20% of the time in a span of 3 months. This is significant because there's a huge cost associated with this. Despite the ability to prevent major amputations, there's about a 50% rehospitalization rate post-revascularization. And what you'll find is that about 50% of those are driven by wound-related issues. infection, need for amputation, and pain. So what we've been able to do and move forward on is what we call the Phase 2b STOP PAD study. So this will be in up to 120 patients. It's a double-blinded, randomized, placebo-controlled study. It builds off a Phase 2a study in which we were able to show that in a similar patient population that was considered no option, we're able to restore blood flow, close wounds, and reduce pain. In this study, we'll focus specifically on a population post-revascularization. So they'll all have undergone a stenting or balloon procedure. But through the measure of what's known as a toe brachial index, we can assess whether they continue to have poor perfusion of blood in the foot. If they do, they will then get injected with either placebo, 8 or 16 milligrams of JVS 100. It's a simple 5-minute office procedure in the ischemic foot. We'll follow those wounds for a period of 3 months and then come back, re-inject the patient, and reassess the wounds at 6 months. We're nearly done with enrollment. Anticipate that'll be done in the next couple of months. We anticipate data from this in the second half of this year. We have been granted fast track designation by the FDA, and I think important to anybody who's followed this field, they've also agreed with us having a registration endpoint of wound closure, which makes the Phase 3 much more manageable and addressable for a company of our size. So as a quick summary, JVS 100 is really a factor-based strategy to activate stem cell-based repair of tissue. We have excellent safety and preliminary efficacy data across multiple clinical indications. We're in the middle of a Phase 2b study for patients with critical limb ischemia and anticipate top-line data to be read out in the second half of this year. And building off that, we truly believe there is a product platform potential here going to areas like heart failure, incontinence, dermal scarring, and others. But happy to take any questions, Josh.
Terrific. So, we hear a lot of the gene therapy companies working with viral vectors, and I guess you've decided not to. What is it about the construct that enables, I guess, kind of is a naked plasmid delivery? And
Yeah.
Maybe elaborate on that.
Yeah, so I think, the vector being a delivery system has to be tailored to what your therapeutic need is. What our early studies showed was that you needed to upregulate SDF1 in a localized fashion for a period of 3 to 4 weeks. There was no added benefit from constitutively expressing SDF1, nor is it necessarily a gene that we want systemically expressed, right? So that's why we focused initially on a non-viral strategy. Now, several people asked us, well, based on that non-viral strategy, why not use liposomes or some other means to increase transfection efficiency? 2 answers to that, which we learned early on in our process, was, first of all, the fact that it's a 6 kilodalton secreted protein, we don't need to get it into 95% of cells. If we can upregulate it to a level above baseline, that's sufficient. The other, and I think most important component about this, is we did test some of those factors. An ischemic muscle takes up naked DNA very well. So in an ischemic setting, post-injury, we end up getting very nice gene expression from a non-viral strategy.
And can you elaborate a little bit on the biology in your comment that you only want localized expression for 3 to 4 weeks and not longer?
Yeah, and probably, that's all we need. So what we know is that if you directly deliver JVS 100 or SDF1 in general to the injured tissue, you recruit stem cells to that, so we think of it as an SOS or a beacon. You also have a direct trophic effect on those cells. So you'll have hibernating cells or cells that are pre-apoptotic, and you have the ability to rescue those. If you systemically deliver SDF1, SDF1 and its axis are very important in holding stem cells in the bone marrow. And what you'll end up doing is you'll release those stem cells from the bone marrow, but they'll no longer have a signal to migrate to the injury. So you'll have circulating stem cells, but you won't have the SOS signal of where they need to go.
Interesting. Maybe you can share a little bit about the experience in heart failure and what you learned there to inform your peripheral artery disease plans.
Yeah. So what I can tell you we've learned through all our programs is that SDF1 does a very nice job of growing blood vessels. And that's primarily what led us into PAD as the lead indication. We did run a Phase 2a clinical study, or an exploratory Phase 2 study in patients with ischemic heart failure. These were patients who are on average 11 years removed from their heart attack, NYHA class 3, so these are patients who are really on that bridge to transplant path. What the data demonstrated to us was that we had a strong safety profile, and we identified a patient population of patients with high cardiac dysfunction, so low ejection fraction, that responded very nicely to the drug. The data itself is exciting. You know, what we learned from that study was we have a population in which we have benefit, but prospective study in that area costs $45 million in 2.5, 3 years to complete. For a company at our stage, what we thought made more sense was go after the near-term inflection point, demonstrate through a phase 2b that we have clinical efficacy, and definitely look forward to coming back and elaborating on that heart failure data at a later point.
And can you share a little bit more about what you saw in the first clinical study in peripheral artery disease?
Yeah, sure. Thanks for asking that. So we did a 48-person study. It was a first-in-man in CLI for JVS 100. So these were no-option patients, meaning they had failed all revascularization. They received in that study one treatment, and we followed them for a year. Great safety profile. Really, out of the 250-plus patients, we've never had an adverse event related to a drug. What we saw were clear trends towards improvement in 4 important areas. We saw wound closure. We saw a reduction in pain. The ischemia creates tremendous pain for these patients, and we saw that reduce over the course of the year. And what that correlated with was improvements in quality of life and improvements in what's known as a Rutherford scale, or grading of the CLI. So really, similar to what we saw in our population of heart failure patients who benefited, the comfort comes in that all the efficacy parameters pointed in the same direction and was correlating with the biology that we believe was in play.
Any questions in the audience? And if not, keep going. I guess as you get to the end of the phase 2b, given just such a huge unmet medical need in this population, lack of alternatives, and potentially a well-run placebo-controlled trial, what are the next steps?
Yeah, so next steps, obviously, from a development perspective, is we're already starting to think about the phase 3. One of the nice things about this indication is it is a phase 3 with a wound healing endpoint that is manageable for a small company, but it's also of great interest for large companies because the market's significant enough. So we'll explore all options in terms of potentially partnering this out, obviously thinking about how to finance this to push the phase 3 ourselves, and then also using that data to really start pushing the expansion of that platform into other muscle disorders like incontinence that we've looked at.
I guess I'm wondering, depending on the signal, is that potentially a registration-enabling study, or if not, why not?
The phase 2b or the phase?
The phase 2b.
We'll have to wait and see the data. I mean, the reality here is when we presented this data to the FDA, I think the data around the need for something to improve foot perfusion, patients having undergone revascularization, the biologics fields in CLI was stuck in no option with amputation-free survival as the endpoint, right? Very hard population, very hard endpoint. What was encouraging to us was through the discussions with the FDA, they saw this huge unmet clinical need to be able to close these wounds. And I like my foot, I like my toes too. I want to keep all of them. And so with that, we did receive the fast track designation. Obviously, if the data is sufficiently compelling, we'll engage them to see how aggressively we can push that towards approval.
And then maybe a quick comment on manufacturing and scale up for commercial readiness.
Yeah, this is the one place I definitely like to talk because our situation is a lot simpler than a lot of the folks here. This is a microbial cell bank, highly scalable, cheap cost of goods. We've already transferred the process to 2 commercial scale CMOs, easily transferable. The nice thing about this is being based out of E. coli, you can make commercial scale product for large indications at dollars per the dose. So really, when we think about ourselves relative to some of the competitors in the cell therapy space, what we have is a product that's cheap, highly stable, easily distributable from a logistics perspective, but has the same impact as some of those cells may.
Right. Well, thank you very much
Thanks,
for sharing the story.
Appreciate
Thanks.
it.